All right, so let's look at the diversity of flukes that are out there. As their most recent classification goes, there's 10 whole orders of them. We're not going to focus on them all. There's 4 that have species that are of major medical or veterinary importance. These are the ones that you're likeliest to encounter if you don't end up becoming a specialist in parasitology. The one that I want to cover today is the order Strigeiformes, important from a medical perspective because they include Schistosoma, not a humongous problem in the United States, but globally a very large public health concern. It includes other blood flukes as well. Echinodermatiformes includes the liver fluke. Opisthoraciformes and Plagiorchiformes includes, I think Plagiorchiformes includes the lancet fluke. And there's various others that we'll encounter that can parasitize humans and also parasitize animals of importance to humanity. You can always tell the Strigeiformes because the cercaria have a forked tail. This is the group that's responsible for most cases of swimmer's itch, which I've talked about. It's that thing where the cercaria burrow into human skin. They don't get very far, but they cause an allergic reaction, causing a skin rash. And I just found out this morning, it's usually not considered serious enough to require medical attention. It's the kind of thing that will clear up on its own in a couple of days, maybe with a little bit of lotion or antihistamine or something like that. But it actually has drawn attention because it seems to be increasing in frequency. It's a so-called emerging disease. I came across a paper pointing out that one of the species that causes cercarial dermatitis, this thing called Trichobilharzia, is distributed on several continents because the adults are parasites in migrating ducks. And so it tends to get transferred from one continent to the other. Some of the hosts that it's in tend to migrate very long distances indeed. And again, there are groups that have flagged it as something we may need to pay more attention to as an emerging disease. Within that group, strigioids, aside from cercarial dermatitis, they don't infect humans. They're pretty common parasites of wildlife. If any of you go on to careers with the State Department of Game and Fish or any kind of careers in wildlife conservation, you might encounter strigioids again. There's a number of very common species that are parasites of water birds, so ducks, also herons and egrets and things like that. And then the schistosomatoids include the human blood fluke. The human blood fluke is unusual because it's got separate sexes. There are separate males and females. Most of the time in flatworms, they're hermaphrodites, so this is kind of weird. And the adults mature in the circulatory system of the host. These don't hang out in your gut or your liver or your bile duct or anything like that. They hang out in large blood vessels where they can cause damage to various organs. Schistosoma was first described by a guy named Theodor Bilhaus back in 1851. And because of this, you'll still sometimes see the disease called bilharzia. That's an alternate word for schistosomiasis, which is the name for infection by schistosoma. You might encounter that term if you go on to any further work in public health or anything like that. If you have to read older papers, you may still see it called bilharzia. Okay, so strigeids don't normally infect humans except for swimmer's itch. This is a common one that I've actually seen in Arkansas. This is uvulifer. The mouth is up at the top. You can see the oral sucker and the ventral sucker up at the top. The mouth leads into that blind gut, which is shaped like a Y. That's typical for flukes. Just about everything in the back half of the animal is reproductive organs, with the two testes in the middle and the female organs kind of snaking around behind them. The gonopore is at the rear, as opposed to the fasciola liver fluke, where the gonopore is up at the front of the body. And adults live in fish-eating birds. Kingfishers, for example, tend to be host for adult uvulifer. I've mentioned the life cycle before, and if everything goes well, the lab we're going to start, not this week, but next week, we're actually going to go looking for them, because it's not too uncommon to find these when you sample freshwater fish in Arkansas. The adults live in fish-eating birds. Eggs usually pass out in feces, penetrate into snails, go through their typical life cycle. The cercaria leave the snails and burrow into fish and form metacercaria. And the metacercaria look like this. The metacercaria burrow into the skin. They don't seem to do that much harm to the fish, but the fish will secrete melanin around the metacercaria as part of its immune response to them. This creates black spots on the outside of the fish. This is called black spot disease, sometimes called black grub. So you can see what it looks like up at the top. On the bottom, that's an actual black spot. We took a picture of that in parasitology class, not this past spring, but maybe the previous fall or something like that. There is a little baby worm kind of snuggled up inside that black football-shaped cyst. And then, of course, if a fish-eating bird eats the fish-eating bird gets infected when the metacercaria break open and out pop the worms. And so this is something you see rather a lot. It's pretty common in local freshwater fish populations. I was hoping I could find the cercariae in the lab that we did last week and this week, because they're common enough as black spots in the fish that I was hoping we could get cercaria out of the local snails. It didn't work very well last week. We'll see how it works this week. I'll try to visit a couple of different places where we might have better luck. So we'll see how that goes. And if it works, it works. If it doesn't, it's like, finding fish parasites is kind of like finding fish. Some days they're biting and some days they ain't. Anyhow, moving along. Schistosomatidae are the blood flukes, and we'll focus on the 5 species in genus Schistosoma that parasitize humans. Traditionally 5, some of them are probably groups of cryptic species. I'm not sure of the current status on that, but you can see that Schistosoma japonicum, despite its name meaning from Japan, is actually prevalent in China and the Philippines. Over to the right, those pink areas, China, the Philippines, and then that's the island of Celebes in part of Indonesia. Schistosoma mekongi is found only on the Mekong River, which looks to be on the border between Cambodia and Burma. That's that green spot on the right. And then Schistosoma hematobium and Mansoni and intercalatum are primarily parasites in equatorial Africa. Some of them getting up into Egypt along the Nile River. Hematobium is found, that's the Tigris and Euphrates in Iraq. And Schistosoma mansoni was introduced to South America and the Caribbean by the slave trade. So those green areas at the left in Brazil and Venezuela and on the Caribbean islands are results of the trade in African slaves. Evidently, when the captured Africans started excreting eggs, they hatched into larvae that were able to live in the local South American snail species. That might be interesting to look at because I assume there would have been a host switch that the larvae would have had to mature in South American snails instead of African snails. Interesting to see how they did that. Right. The life cycle looks like that. You're infected by contact with water containing the Cercariae, and the Cercariae burrow directly into skin. The Cercariae will transform into adults. It takes about 6 weeks. They'll enter deep blood vessels. They particularly seem to like the portal circulation, which goes through your liver. They can end up in the blood vessels of the bladder, the spleen, the gut, the mesentery, anything like that. They'll mature there, and they'll pair up because this species has separate males and females. And they'll start producing eggs, and the eggs work themselves out of the body, and they pass out in urine or feces. There they hatch into myricydia, penetrate into snails, develop into Cercaria larvae, and the Cercaria burrow into new humans, and it's the circle of life, and it moves us all. So there's a pair of the adults. The male is the larger one. Most of what you can see in this picture is the male. And up at the top and a little to the left of the picture is the mouth, and you can see the ventral sucker of the male as well. And then just past the ventral sucker, there's something that looks like a hose sticking out of the male. That hose is actually the female because she lives inside a long, narrow groove on the male's body called the gynecophoral canal. Right? Gyneco meaning female, phora being Greek for to carry. So this is the woman carrying canal. Basically, they spend their entire adult lives spooning. You can see the male's got fairly prominent suckers. The female, they're highly reduced, although you can kind of see one, the tip of the female's body, which is down at the lower left. The male is also covered in bumps called papillae, typical of a man's eye. And there's what they look like snuggled up inside a blood vessel. And different species prefer different niches. Mansoni prefers the large intestine. Hematobium prefers the bladder. Japonicum prefers the small intestinal veins. Not sure how they know where to go, but there you have it. They have different preferences. By the way, you may already know this, but in case you don't, the circulatory system you usually think of as blood leaves the heart. It goes through arteries, it goes through the capillaries, it goes through veins, and then it goes back to the heart. The exceptions are when you have blood that goes through arteries, goes through capillaries into veins, and then from the veins, it goes into capillaries again, and then into another vein. So if blood that is traveling back to the heart through veins takes a bypass and goes through another organ in a capillary bed again, we call that a portal circulation. And probably the best known portal circulation is the hepatic portal circulation that goes through your liver. What happens is that arteries go to your intestines. They pick up digested food as well as feeding the intestines with oxygen. And then with this digested food, instead of going straight back to the heart, they take a detour through the liver and pass through the liver, and once they're out of the liver is when they go back to the heart. So that bypass via the liver is called the hepatic portal circulation. And this is important for understanding what schistosoma does. The worms are in the vessels and they start laying eggs. And last I looked, it's not entirely clear how the eggs do the trick. But they pass through the walls of the gut or the walls of the bladder and pass out in feces or in urine. There aren't a whole lot of parasites you can get from urine. This is one of the few. And you might need to know this if anybody goes on to public health. The egg shape is diagnostic in different species. Schistosoma mansoni has a spine that's off at an angle. That's the one you see at the top, the upper tip, that's a side. Spine at the tip, spine, that's the picture at the bottom. And there's no spine for Schistosoma japonicum. And as you'd expect, the eggs hatch into myricydia. The myricydia infects snails, and they form sporocysts, and the sporocysts form daughter sporocysts, and the daughters form cercaria. Remember that, I showed you the life cycle of the common liver fluke, but mentioned that you can have stages that get doubled or deleted. And this is one of those cases. There's no readiest stage. Daughter sporocysts directly form cercaria. The host of Schistosoma mansoni are snails in a genus called Biomphilaria. Here's a Biomphilaria, and all those little white flecks surrounding it are cercaria. This is what a snail looks like in the act of shedding cercaria. This is what we were hoping to see last week and didn't, but maybe this week will go better. And there's a single cercaria. The technical name for a cercaria shaped like this is Furca-circus. Furco-circus means got a tail. Furca means anything that's forked. Frankly, I think Furca-circus is a pretty good word just because it sounds like a word you might use to, describe modern life. Between the pandemic and the forest fires, the world is just a Furca-circus. Go ahead and use that if you want to. It sounds really apt to my ears. Anyway, these penetrate to human, shed the tails, migrate to the bloodstream, mature in the liver for 3 weeks, and then migrate to their preferred position, wherever it is. In the Asian species, Schistosoma japonicum, getting infected brings a low-grade disease called Katayama fever. In the African species, I don't think there's an awful lot of symptoms when you get initially infected. So the adults lay those eggs, and the eggs migrate through the tissues. This is a picture. This would be liver tissue. And in the center, that thing right in the middle that the arrow is pointing to, that's actually an egg. And you can see it's kind of in an open space, and surrounding it, you've got a lump of scar tissue called a granuloma. And I'm not sure how they do it, but the egg actually seems to need the granuloma in order to get through the liver. It may be that the immune system has some way of picking up the egg and pushing it out. And we think that because in immune patients, they suffer a lot more liver damage from having eggs laid in the liver, but they don't excrete them efficiently. So there's something about that immune response, that granuloma, that the parasite actually needs in order to get its eggs into the outside world. Now, the granulomas do protect the host by blocking the eggs off from healthy tissue. At the same time, the eggs are somehow able to take advantage of granuloma formation in order to get pushed out of the organ that they're in and get into the bile duct or the bladder and go on into the wide world. So kind of neat there. Interesting strategy for trying to develop treatments for this might be to try to figure out how the egg is moving and then see if you can find something that might block it. The problem is that if those granulomas block circulation in the liver, if they start blocking liver capillaries, they cause swelling of the liver and spleen. This is called hepatosplenomegaly. If it's only the liver, it's hepatomegaly. If they're both involved, it's hepatosplenomegaly. And going with that is portal hypertension. That's what it sounds like. It's high blood pressure within the liver, within that portal circulation that takes blood from the intestines through the liver and then finally into the vena cava and back to the heart. That is potentially life-threatening. That will cause permanent liver damage, to say nothing of the swollen abdomen that you can get. Here's a couple of rather dire-looking cases of swollen abdomen caused by heavy schistosoma infection. The next slide's even worse. Brace yourself. Schistosoma tobium hangs out in the blood vessels surrounding the bladder, and the eggs work their way through the bladder wall and end up being released in the urine. This causes damage to the bladder wall, the most obvious form of which is blood in the urine. This, by the way, is a common species in Egypt, and it was actually described in some ancient Egyptian medical documents. They had medical books back in the day, and this was the kind of thing you could get if you spent too much time splashing around in the Nile River. If memory serves, the name for it literally transliterates as "ah." I'm not even kidding. That was the word for this this particular infection. There are parts of Africa where one of the things that men have to take care of is maintaining irrigation ditches, which means that men will spend, hours every day wading in irrigation ditches. So, of course, if there are snails in the ditches, which there always are, the men get infected with cercaria. Right about the time boys hit puberty happens to be the time when boys start helping their fathers at work and start going into the ditches themselves, which means that boys start getting bloody urine at roughly the age of 12. And these groups basically consider it to be menstruation, that, women start bleeding from their bits at about the age of 12. So do men. They accept it as basically a normal part of life, when in fact it's parasite infection. And not one that you'd really want to get. I mean, aside from peeing blood, granulomas in the urinary tract can narrow the urinary tract or even block it, which is really not something you want to have happen. They can harbor bacteria and lead to a secondary infection. And they are associated with a higher risk of bladder cancer later in life. So, yeah, this is why this is a potential public health hazard. You really don't want this to happen. In 2010, there were an estimated 238 million people infected with schistosoma, 85% of which lived in Africa. 112 million people each year get infected, and this is linked to 150,000 deaths annually, mostly from kidney failure. Non-fatal infections can still cause various illness. They still cause lethargy. If your body is dealing with a sizable parasite infection that drains energy from other things that you're doing. If you get it in childhood, it can even stunt your growth. And if women get Schistosoma hematobium, granulomas can affect the female reproductive tract, and that can increase the risk of HIV transmission. And HIV remains a severe public health problem in many parts of sub-Saharan Africa. So completely aside from the loss of life, Schistosoma has a pretty bad economic impact. So what can we do? There is a drug, praziquantel, that can treat this. There's a vaccine in development. Last I looked, we didn't have the results from phase 3 trials. As you know, because it's in the news all the time, it takes a while for vaccines to be brought to market. And, we may not get a vaccine for COVID for 18 months after it's first detected, and that's with multiple governments throwing everything they've got at the problem. Schistosoma tends not to register on the radar quite so much, so it probably will take years before a vaccine's ready, assuming that it's effective. As with a lot of things, improved sanitation and waste disposal are probably the simplest means. Peeing and pooping in places where the eggs can't hatch into larvae that infect snails is probably the single simplest thing that can be done to rein in Schistosoma. Telling people, don't pee in the Nile River, things like that. In some areas, molluscicides have been used, chemicals that kill snails. They can have problems of their own. However, they may kill other things as well. They might have an impact on snail-eating fish populations or something like that. Trying to treat a parasite infection by poisoning the local ecosystem certainly very often will have downsides. They've tried some introduced snail species that can outcompete the native snails. Usually you don't want introduced species that outcompete the natives. I guess Dr. Larson has retired, but if any of you knew her, that was her big thing. She studied introduced species, plant species in her case. But this is a case where it's been tried and seems to have worked to reduce the incidence of Schistosoma. The other thing is that snail populations can explode wherever you have slow water flow. Most snails don't do particularly well in fast-moving water, right? They get picked up off the bottom and dragged away. Water flow might pick up the vegetation that snails would eat too. So this is not much of a problem in whitewater areas. But in places where water doesn't ever dry and where the flow is very slow and limpid, snail populations can get quite large. And this is places like irrigation canals and especially lakes behind dams. Dams can, because they create slow flow, lots of slow water habitat, they can boost snail populations. Dams can also decrease the population of would-be snail predators, such as freshwater shrimp. There are parts of Africa where they've tried introducing snail predators, notably Procambarus clarki, which is the edible crawfish from Louisiana. To a crawfish boil, you were eating Procambarus clarki. Apparently introduced only African populations a very delicious food source, but they've succeeded in reducing infection rates. So there's a drug, there may be a vaccine, there's a number of things you can do, and this is something I keep coming back to, is to treat parasites. Often the simplest things you can do and the quickest things you can do are trying to cut the life cycle at a weak spot And controlling the snails is one way of trying to do that. Classic example of unintended consequences. That's the Aswan High Dam. If you know your ancient history, you might know that the rhythm of life in ancient Egypt was that every spring, the water level in the Nile rose, and the Nile would overflow its banks. When the river went down, it would leave a new layer of silt over everything, which is what made Egypt very fertile. You know, as long as you're right next to the Nile, the fields were very fertile. You know, get very far away from that and you're in the desert. And the wet season, when the Nile had overflowed its banks, was when the farmers couldn't work. So they'd go get jobs building pyramids or hauling statues or things like that. Those big, you know, the pyramids and other monuments weren't built by slaves. They were built by farmers, you know, who were free and who were paid, but whose labor was required during the time of year when they couldn't be working in their fields. Anyway, that cycle of life came to an end with the building of the Aswan High Dam, so the Nile doesn't overflow anymore. And it made water available for massive irrigation projects. So even though you don't have the natural ebb and flow of the Nile, there is plenty of water available for irrigation. Unfortunately, the Aswan High Dam built Lake Nasser behind it. Gamal Abdel Nasser was the ruler of Egypt at the time. I forget if he was a president or a premier, but he was in charge. And the irrigation canals and the lake behind the dam turned out to be optimum snail habitat. The snail population exploded, and infection rates in the Nile Valley went from about 5% before the dam was built to about 35% after the dam was built. That's an increase of 7-fold. So, yeah, one of the unintended consequences of building the Aswan High Dam was a great increase in Schistosoma. Some of the things you can do with a dam project might involve controlled releasing of water through the dam. If you can open up the dam just long enough to let some water drain, what that can sometimes do is create fast enough current flow that it can sweep away building any snail populations that are building up. So there are things you can do to mitigate. All right, and we're ending early, but that seems to be what I've got for you today. So I will go ahead and pause the recording.